Hi, science friends. I'm sorry that I can't be with you all today. I'm doing well, but I wanted to not miss this opportunity to talk about DNA replication. So let's try to dive into just a little bit. We will go over all of this again because it's pretty in depth, but I wanted you to be able to start working on a replication diagram today. So, DNA replication follows what we call a semi-conservative model. And so what that means is that in the you've got the original strand of DNA, the parent strand, and it's going to form two daughter strands, just similar to mitosis having the parent cell that forms two daughter cells. But the semi-conservative model of replication means that the new DNA has one half of it that is part of the parent strand, and one half of it's part of the is the new strand. So here we've got our original parent molecule. It splits in half. The new daughter part is created for both of those parts. They're filled in here and filled in here. And so the daughter DNA has one of the originals, that's the dark blue, and then one of the new strand. And so we call that the semi-conservative model of replication. And so this diagram just kind of shows the experiment of how that was done, looking at bacteria that were radioactively labeled, similar to the way that they determined DNA as the genetic material. They tested a few different models, like the conservative model where you had all the parent DNA and then the whole thing was copied and you just got a whole different set of daughter DNA. The semi-conservative model, which we know to be true, half of it's the new and half of it's the old. Or a dispersive model where you kind of have pieces of old and new interwoven together. And so what we do know to be true now is the semi-conservative model of DNA replication. Okay? So when we start DNA replication, remember DNA is in a double helix shape. I don't think we actually used that term earlier this week when we were talking about the structure of DNA, but the double helix is the shape of the DNA molecule. It's the spiral or twisted staircase. And so what happens in that is in the middle of the DNA molecule, the DNA opens up. Okay, it kind of unzips in the middle. So for example, like on your all's backpacks, most of your backpacks, the biggest pocket has two zippers, right? And so maybe some of you take it all the way, your zippers all the way from one side to the other. But some of you, I know, take one zipper from the left and one zipper from the right and kind of zip it up and meet those zippers in the middle. And so that's kind of how DNA replication opens. If you've got both zippers together at the top and the middle and you pull them apart, then you have just a small portion of your backpack open and you can reach down and grab your phone or your earbuds or whatever. Okay, and that's how DNA replication happens. The zipper opens and there's just a little piece that gets replicated at a time. And that area that it gets opened up is called the replication fork. Okay, so if we're looking at our DNA, let me try to draw this as best as we can. Okay, so this area here is referred to as the replication fork. It got opened up in the middle. Okay, here and here is the origin of replications. That's essentially where you pulled your zippers open at. The origin of replication is where the replication begins. So when replication happens, it is going to happen in two directions. Okay, it is going to go in one way on one strand and in the other way on the other strand. Okay, now remember the term that we used yesterday called anti-parallel. Okay, so if this top strand, if this is 5 prime, then that means that this is 3 prime, and that means on the opposite strand that this is 5 prime and this is 3 prime. And so DNA synthesis, notice the way that I wrote these arrows, is always going to happen, or replication, from the 5 prime to 3 prime direction. Okay? DNA replication is always 5' to 3' direction, okay? And that's very, very important. Now, in eukaryotes, because the DNA is so large, there's sometimes multiple sites of DNA replication, okay? So if you've got your big, huge DNA molecule, you might have one replication fork there, and another replication fork there, and another replication fork there, okay? So DNA might be copied at several different times in several different spots at once, okay? And that just makes it more efficient, makes it able to copy the DNA faster. Now, in prokaryotes, you're going to only have one origin of replication because the prokaryotic DNA is smaller and it copies itself faster in general because it's smaller, and so there's only going to be one origin of replication site there. So that's something that's just a little bit different between prokaryotes and eukaryotes, okay? But we get this replication fork they call it a Y-shaped region, so you can kind of see the sideways Y there, where the DNA is unwound, and then the new DNA is synthesized. So this here is going to be the parent strand, and this would be the parent strand also, and then new DNA would be made in here, and those would be the daughter strands, okay, to show the semi-conservative model of DNA replication. Okay, so this diagram just does it a little bit prettier than I did. So here we've got a eukaryotic cell, and you see those multiple different replication forks. The origin of replication is in the top and bottom, kind of in the middle of each replication fork, or sometimes they're referred to as a bubble. The template strand, which is another word that's sometimes used to describe the parent strand because it's giving us the DNA template, is the part of the original DNA. And then the light blue here is called the daughter strand or the new strand. That's what's being synthesized new. Okay, so that's in our eukaryotic cell. Now, this shows E. coli, so E. coli is going to be a prokaryotic cell. Bacterial cells, they have different, DNA is a little bit different. So their DNA is a circular molecule, and that's called a plasmid, okay? A circular DNA molecule. It's still double-stranded, but they only have one origin of replication, and so it's going to happen in both directions until they kind of meet in the middle, and then you'll end up with the two new daughter molecules that are shown here, okay? So little differences there again between the prokaryote and the eukaryote cell. Now, in DNA replication, there are a ton of enzymes that you have to know, okay? If you feel like you are a flashcard maker to study, this would be a great time to make flashcards, okay? We're going to talk about probably 11 or so enzymes today, and there's a couple of those that I'm not going to necessarily require that you commit to memory, but many of them you will need to commit to memory, okay? So some of them you learned about a little bit in the video that you watched yesterday on the Amoeba Sisters. I hope you liked that. I always think they have good videos. That's a great place. They have a whole website with videos, a YouTube site with videos. So if you're ever struggling with a topic, that is a great place to go to just try to get a quick overview and review for a test, okay? So enzyme number 1 is DNA helicase or helicase. Either way is appropriate to say it. And I think the amoebas called it the zipper, okay? Or rather, the unzipper. And so DNA helicase is responsible for opening up that replication fork. It is the zipper on your backpack. It is what creates that replication fork and separates the two DNA strands, okay? That is helicase. Now, another enzyme or protein rather, that is important is called the single-stranded binding proteins. Okay, so what they do is where that replication fork opens up, they are gonna kind of bind right here and help to hold that open. Okay, helicase is going to unzip it, but we have to have somebody that helps to hold that open. And so that's going to be the job of the single-stranded binding protein, okay, to keep it from opening back up. Now, another enzyme, many of these are enzymes because they're going to end in ACE. All enzymes, of course, are proteins, is topoisomerase. And so topoisomerase is also going to be acting in here in the still coiled DNA, okay? So what's going to happen when you open this section of DNA up is it's going to cause this coil to get even tighter, okay? It makes that coil tighter there on those ends. I don't know if you can envision that, but it makes perfect sense to me. I can kind of see it happening in my head. If you open this up and you create a gap or more space in that area, that's going to cause this area to coil tighter, okay? And so what topoisomerase does is it actually comes in here and undoes some of the hydrogen bonds between the two strands and unravels it a little bit and then reconnects them back together. And in doing that, it helps relieve some of the strain on the DNA. So it breaks that hydrogen bond, it swivels it to unravel it a little bit and loosen up that coil so it's not quite so tight anymore, and then it joins them back together so it's one whole DNA molecule again. Okay, so it's actually working on the parent strand there. Oh, I gotta get it together, finish up before my battery dies. All right, the next one is DNA polymerase. Now, there are hundreds of different types of DNA polymerase. We are going to learn about 3 of them, okay? So, the first one let's talk about is DNA polymerase 3. And so what its job is to add new nucleotides, okay? It's perhaps the most important of the enzyme. So if we are reading along here on the parent strand of DNA, and this is our code, and it's reading GCTAGG, it is going to get the new nucleotides. If it reads G, it's going to go get cytosine and bring it in to pair it up according to Chargaff's rules. Remember those from the other day, okay? So it is continuing to add DNAs complementary to the template strand or the parent strand. So it is super important, okay? Oops. Maybe. Now, primer, okay? Primer is an RNA segment, okay? I think you heard about it with the amoeba sisters as well. It tells DNA replication where it's going to start. It tells DNA polymerase where to start. It is the starter, or I think they called it the initiator. It has to bind first, and it lays down a section of RNA primer, okay? The enzyme primase lays down the RNA primer, okay? And that RNA primer is where polymerase 3, is going to bind to be able to start the new DNA replication, okay? Here you see those single-stranded binding proteins standing here in the Y to hold that open. You see the topoisomerase. Notice that he's kind of broken that bond there and is going to you can't see the helix there, but he's going to unwind that a little bit to relieve some of the strain. Okay? This is an important term, nucleoside triphosphate. So that is basically the new bases that are coming in are called nucleoside triphosphates. Here's our template strand. We're reading TGCAC. And the triphosphate is bringing that new base in to connect and replicate the DNA, okay? Job of DNA polymerase, okay? Now, remember our nucleotide is called a sugar phosphate base. So here we've got our sugar, here's our base, and here's our phosphate. So the triphosphate, we're going to drop these last two off and just leave our nucleotide that's intact, okay? So nucleoside triphosphate's what's coming in, and then you're going to recycle these P's. They're going to leave, okay? That's not super, super important. If you forget about anything, forget about nucleoside triphosphate. All right, again, 5' to 3' direction, okay? 5' to 3' direction. Notice this arrow is going this way. This arrow is going this way. So this is 5', this is 3'. Prime is the little arrow here. 5', or apostrophe rather, whatever you want to call it. 3'. Okay, so and that's the DNA polymerase that's adding the nucleotides in that order. Okay, again, we see all of our enzymes over here. The primer is the segment of RNA. It's shown as red because it's not DNA. Our polymerase that's bringing in the new nucleoside triphosphates. Our origin replication is where the primer started here. Okay, now one thing that you may have noticed in this diagram is how these arrows are several little short arrows, as opposed to this arrow is one nice long continuous arrow. Okay, and so the one nice long continuous arrow is called the leading strand. The lagging strand are these short arrows. Okay, notice that they still have that primer that starts them, that RNA primer that is synthesized by that enzyme primase. Okay, so leading strand, it is created as the replication fork progresses. So wherever the origin starts, it's going to be the one that's going from 5' to 3' from the start of the origin. Okay, same over here, from 5' to 3' from the start of the origin. Only one primer required for it. We see only that one piece of primer that was required there, and it's one long continuous segment. Versus the lagging strand is moving away from the replication fork. It's going back in here where the DNA is still coiled up, okay? It's going in the, back in here where the DNA is still coiled up. And it's going to have a primer for each of those fragments. And these lagging strand fragments are sometimes referred to as Okazaki fragments, okay? That is a term that you often hear called that. And one that I will never, ever forget my Middle Eastern professor, Okazaki fragments, saying over and over and over. He loved to talk about the Okazaki fragments in molecular biology. So when you hear that term, they're just talking about these little short lagging strand pieces, the Okazaki fragments. Okay? All right. Now, let's get into our next polymerase. The first one that we talked about was polymerase 3, okay? It was bringing in those nucleotides, those nucleoside triphosphates, technically, okay? Polymerase 1, we use Roman numerals here. It is going to replace the primer, so we've got our red primer here. It is going to replace it with nucleotides. Again, still going from the 5' to 3' direction. If we're going to copy this DNA, that's what we're doing here, DNA replication in interphase. Before a cell divides, we have to have a whole smooth strand of DNA. We can't have these little Okazaki fragment chunks, and we can't have these little primer chunks. And so RNA polymerase is going to replace the primer chunks, okay, with another fragment piece, okay? And then eventually they are going to all be glued back together by the enzyme DNA ligase. Okay, the Okazaki fragments are going to be sealed together by DNA ligase, and once the primer is replaced with DNA, it is going to be sealed together and make one smooth, continuous piece of DNA. Okay, by joining those sugar phosphate backbones. And again, remember that is a covalent bond that holds those sugar phosphate backbones together. All right. So again, here's a picture that just kind of shows all of what's going on. Primase is putting in the primer, the RNA primer that's red. Polymerase 3 is bringing in the new bases to make the new DNA. The lagging strand is these little chunks, okay? Polymerase 1 is replacing the primers. Ligase is gluing all the way back together. Here's our nice long leading strand. Single-stranded binding proteins holding it all open in the replication fork. Polymerase 3 again here bringing in those nucleotides. This guy would be topoisomerase, helping to relieve some of the tension on the tightening of those coils in the replication fork. Okay, so lots of really important people. So what I want you to do today is you have this diagram, okay? You should have this in your binder. Oops. It is about 3 pages long. It is 3 pages long. So you'll want to cut them out and tape them all together. Over on my side table, there's scissors and tape and colored pencils. And I want you to make it pretty, okay? This will be one beautiful diagram. And if you take biology again, you will learn about this again. And you will pull out this diagram and you'll be like, dang, I'm really glad I kept this, okay? So I want you to revisit each of these enzymes, okay? Like, for example, number 1, let's find it. Number 1 is right here. It is unzipping, okay? So if you remember our unzipper, number 1 is helicase. So you tell me what helicase does. Enzyme that unzips to open up the replication fork, whatever, okay? And color that in there, give it a color here, okay? If you make a nice, pretty diagram, you'll be glad you did. It will help you study it better, and you'll walk away with a better understanding of replication, okay? Number 2 is going to be topoisomerase, all right? Number 3 is the single-stranded binding proteins. Okay, that's these guys here that are holding that replication fork open. If we scroll down to number 4, what is this guy doing? He looks like he's building the primer. That's going to be RNA primase. That's number 4. I think that's the arrow for number 4. Okay. I don't know, I may have cut my numbers off there. But that is number 4. Number 4 is RNA primase. Okay. Number 5, he is bringing in these nucleotides. So that would be polymerase 3. DNA, polymerase 3. Don't call it polymerase. 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 Okay. That's how you say that word. Number 6, those are going to be those nucleoside triphosphates. All right. Let's find number 7. Number 7, okay. I think number 7 is referencing here. That's going to be the leading strand. And then down here we'll have the lagging strand. Okay. 7 and 8, leading and lagging strand. Number 9, I think is this guy. I can't tell in this picture because it's kind of cut apart. But that is number 9, I believe, is polymerase 1. DNA polymerase 1. Okay. 10 is our glue. The glue is ligase. And 11, I don't know where 11 is. I don't see 11 on here. I do not see 11. Okay, maybe there's an 11. If there is, hopefully you can figure out what it is. If not, we'll talk about it when I come back. So, spend some time with this. Make it pretty. Color it. Tell what each of these do. If you get more knowledge later, go back and add it to here. But this is a great, great, great study tool. And I think you'll be glad if you do a great job on it. If you don't finish it, it is not homework. We will continue to work on it and add to it as we go along. Okay? And again, we will revisit all of these things when I'm back. You'll have a great day and a great weekend. Let me know if you have any questions. Thanks, guys.